Thank you for your kind introduction. I'm Atsushi Takata from CBS. So I'd like to show case series of open data utilization in disease genetics and genomics, because this is a case study session. So first, the case descriptions. So when you check our homepage, there is a page for our representative papers, and you can repeatedly find this description. We have only spent the electricity cost except for the initial investment in the computing system. And for another paper, we analyzed the publicly available large genome data. So maybe we are professional in utilizing open life sciences data. And because the time is limited, I'd like to introduce these two cases. The first one is this one. So the title of case one is refinement of the clinical variant interpretation framework by statistical evidence and machine learning. So this paper was published in Med, a Cell Press journal. And a good thing is in Cell Press papers, there is a section for key resources table. So if you are interested in our study, you can check all the public resources used in our paper. And among them, the main resources are shown here. And the main target of this study is this ACMG-AMP, American College of Medical Genetics and Genomics, and the Association for Molecular Pathology guideline. That is a standard framework developed by ACMG and AMP to classify genetic variants based on their clinical significance, according to ChatGPT. And we also used these resources. The first one is Nomado, Genome Aggregation Database. This is a publicly accessible resource that aggregates and harmonizes genetic variation data from general population worldwide. So this is a general population data. And the second one is HGMD, Human Gene Mutation Database. This is a comprehensive resource that catalogs published genetic mutations associated with human inherited diseases. And the third one is ClinVar. This is a publicly accessible database that links genetic variants to their clinical significance and associated conditions. So these are databases for disease-related variants. So first, briefly describing this ACMG-AMP guideline, variant interpretation framework. So in this guideline, the pathogenicity of a variant is assessed based on various criteria, which are categorized into 4 distinct levels. The first one is very strong VS, and the second one is strong, and followed by moderate and supporting. And for instance, loss of function mutations, including nonsense, splice site, or frame shift variants, these are expected to totally disrupt the gene function in genes known to cause diseases are classified as very strong, most strong one, while de novo, newly occurring mutations are classified as strong. And the final pathogenicity of a variant is determined by the combination of these criteria that are met. For example, one PBS very strong and one strong is classified as pathogenic, while one very strong and one moderate is classified as likely pathogenic. And among many criteria in the ACMG guideline, there are some criteria thoroughly defined by the variant types. So as I told, nonsense, frame shift, splice site, and also exon deletion mutations are classified as very strong. And in this PBS category, start-lost variants disrupting AUG or ATG methionine encoding codon is also classified as PBS. And in-frame indel mutations not causing the shift of reading frame in the translation is classified as moderate. And stop-loss variants disrupting stop codon and cause read-through of the reading frame is classified as moderate. damaging missense variants only changing one amino acid is classified as supporting. But I suspected if this is really correct or not. And we decided to use open life science data to inspect this. And specifically, we used this NOMAD, real-world data of variants in protein coding regions from this number of many individuals. And actually, the number of individuals is increasing in the newer versions. And before utilizing this NOMAD data, we performed a forward genetic simulation mimicking the immigration history of mankind, starting from Africa and divided into the populations in Europe with a bottleneck event, and then spread into Europe and Asia. And in this simulation, what we found is the deleterious types of variants showing under the strong negative selection pressures is enriched for rare variants indicated by this dark red. And when we checked the proportions of these very rare variants in established loss of function variants defined as nonsense, canonical splice site, and frame shift variants, and damaging missense variants moderate supporting in ACMG guideline, and all missense, non-damaging missense, synonymous mutations not changing amino acid. As expected, we observed a higher proportion for established loss of function mutations, followed by damaging missense, all missense, etc., etc. So we applied this framework to these three types of variants under suspicion. And what we found is these start-lost variants categorized into moderate showed a high proportion of rare variants. And stop-loss variants showed, inframe indel variants showed low rare variant proportions that is lower than that of damaging missense. And start-lost variants categorized into very strong showed quite low proportion of very rare variants. And we also confirmed that there are highly statistically significant differences across these variant types. So in summary, the pathogenicity of stop-lost variants, this guy, in ACMG guideline might be underestimated. And the pathogenicity of these non-repeat inframe indel variants and start-lost variants might be overestimated in this guideline. So we proposed this kind of repositioning of these types of variants. But only criticizing the existing framework may not be very productive. So we attempted to extract the deleterious variant subtypes using the proportion of rare variants as an indicator. So in the case of stop-loss variants, we found that those causing addition of long abnormal amino acids, say those introducing abnormal 30 or more amino acids, are as enriched as for very rare variants as established loss of function variants. And in the case of inframe indel variants, we found that this protein too small. Variation effect analyzer can effectively extract damaging inframe indel variants that is enriched for very rare variants as damaging missense variants. And in the case of start-lost variants, we hypothesized that if the ATG sequence nearest to the known start codon is inframe, we expected that these might be less deleterious because if alternative start initiated at this site, this didn't cause frame shift. And we expected that this kind of out-of-frame start-lost variants might be more deleterious. And this assumption was correct. we found that nearest ATG out of frame start lost variants showed higher proportion of very rare variants. Next, we can further extract the subsets of start lost, stop lost, or inframe in the variants that are enriched for deleterious variants. And for start lost variants, we further tried to stratify these nearest ATG in-frame start lost variants based on the distance between the known start codon and the nearest ATG, expecting that when the next ATG is close to the known start codon, this might be less deleterious. And if the distance is very long, we expect that these might be more deleterious. But this was not the case. When we analyzed the proportion of very rare variants, we found that rather these variants showed higher proportion of very rare variants. But this may not be totally surprising, because there is accumulating evidence indicating that protein translation can occur at various locations within a transcript. And translation can be initiated from ATG-like sequences, such as CTG, and such translation may occur more frequently than previously thought. So things are very complicated. So next, we asked machine for help. And in practice, we first extracted all potential start site ATG and near ATG sequences and scored the strengths of these ATG-like sequences as a start site by using deep learning model trained on experimental data of translation initiating ribosomal sequencing. And using these scores, we aimed to construct a model to classify known pathogenic variants in HGMD and CLINVR and non-pathogenic variants in NOMAD using these scores and codons and positions and reading frames as features with decision tree based algorithm. And finally, we constructed a model named postal pathogenicity of start loss and checked the performance of this postal model. And what we observed is this side is good performer, and this side is poor performer. So this is postal. So postal far outperformed other tools. And when we extracted start loss variants with a postal score showing more than 95% specificity, these variants are as enriched for very rare variants as damaging missense variants. But actually, there is some trick. So these other tools are not specifically designed to predict the pathogenicity of start loss variants. So our results indicate that the importance of constructing a model optimized for the variant types of interest, or in our case, start loss variants. So this is a graphical summary. So we utilized various open resources and machine learning approaches. We proposed the way to refine the clinical variant interpretation framework based on statistical evidence and machine learning. So this is case 1. Let's move on to the case 2. So the title of case 2 is topologically associating domains define the impact of de novo promoter variants on autism spectrum disorder risk. And again, this paper was published in a Cell-Press journal, and there is a longer list of publicly available datasets used for our study. And among them, we specifically analyzed the data that can be obtained through a SFARI base maintained by Simon's Foundation. And this SFARI base serves as a centralized repository for autism and autism-related research data and biospecimens, along with an online portal facilitating research recruitment requests. And when you push this available resources button, we can see the list of available resources. And among them, in this study, we analyzed whole genome sequencing data. And specifically, we analyzed data derived from autism spectrum disorder patients, neurodevelopmental disorder characterized with impaired communications and repetitive patterns of behaviors and interests, and their unaffected siblings, and their unaffected parents. So this family dataset allows us to analyze de novo variants only observed in the child and not seen in parents. And specifically, we focused on promoter regions because a previous study analyzing the subset of this dataset reported that promoter de novo variants are enriched in autism cases when compared with unaffected siblings. And considering the subset of promoter variants that may contribute to autism risk more, we expected that these immediately upstream of non-ASD gene, say ASD gene linked promoter mutations, may contribute more to the autism risk. But when we tested this, unexpectedly, we didn't see any significant enrichment of ASD gene linked promoter de novo variants in ASD profiles. So we need to think in a different way. And related to this, an interesting study was published several years ago. And in this study, the authors analyzed the effect of promoter deletion by CRISPR-Cas9. And as expected, they observed significant reduction of downstream gene. But simultaneously, they observed increase of many other genes in the same topologically associating domain, where enhancers promoters are frequently interacting within this domain and not across this border. And after performing a series of experimental analysis, the authors finally concluded that when a promoter is disrupted, the enhancer originally coming to this promoter is released and retarget other genes in the same TAT and cause dysregulation of these genes in the same TAT. So according to this, we hypothesized that not only the ASD gene linked promoter variants, but also promoter mutations in TAT containing ASD gene may confer the risk of ASD. So we tested this hypothesis by using public TAT data of prefrontal cortex from psychoencode. By stratifying de novo variants into these three types. And in the reanalysis of published data, we observed some trend of the enrichment of ASD gene TAT promoter variants. And to inspect this, we analyzed openly available data with larger sample size. We observed significant enrichment of ASD gene TAT promoter de novo variants, whereas there is no significant enrichment of non-ASD gene TAT promoter variants. And in the meta-analysis, we found that these ASD gene TAT promoter variants are specifically enriched in ASD profiles when compared with unaffected siblings. So this is the statistical evidence suggesting that topologically associating domain TATs define the impact of de novo promoter variants on autism spectrum disorder risk. And in this analysis, we used prefrontal cortex data. So to extend this, we performed an analysis using tau data from various tissues, utilizing 3D genome browser built based on the ENCODE data, which is, again, publicly available. And in this analysis, what we found is enrichment of ASD gene TAT promoter de novo variants in ASD are pronounced when brain stem cells shown in red letters, TAT data are used. And the top tissues include iPSC-derived neurons, cortical plate, germinal zones. These are immature brain neural progenitor regions. And in interest of this study, we experimentally analyzed the impact of selected ASD gene TAT promoter DNAs by introducing this mutation in iPS cells using CRISPR-Cas9. And when we performed RNA-seq, we found that the two downstream genes, these genes transcribed in this way, two genes are upregulated significantly. And a bit distant neurodevelopmental disorder gene is downregulated, so mediated by some indirect effect. So we confirmed that there is local alteration of multiple genes in the same TAD. And finally, we analyzed the effect of this mutation on transcriptome, and we identified many differential expressed genes. And we found that these differential expressed genes are significantly enriched for non-ASD genes. And moreover, when we performed gene ontology enrichment analysis, we found that these differential genes were enriched for those related to embryo development, neuron projections, etc., etc. So we found that global transonic change enriched for ASD neurodevelopmental genes were observed in these mutant cell lines. And summarizing this story, I'd like to exemplify with this 風が吹けば沖合が儲かる. Let me see this memo. So, if a strong wind blows, there will be more blind man because of the damage to people's eye from the dust. Since blind men play the shamisen guitar for a living, there will be a greater need for cat skin used for the guitar drum. And this will deplete the cat population, and the rat population will increase. Without enough cats to control them, rats cause significant damage to udon items, including barrels. And then barrel makers will do a land office business. So this is a whole story or mechanism of 風が吹けば沖合が儲かる. Like this, in this study, we found that a promoter is mutated. Then local alterations of multiple genes observed. And that lead to global transonic changes enriched for ASD genes and neurodevelopmental genes. And this may cause increase of ASD risk. So this is the summary of our findings. So this kind of phenomenon, like butterfly effect, where butterfly flip in Brazil can cause tornado in Texas, may explain a part of ASD genomic risk factor architecture. This is a press release of a paper. And also, through these cases, we deposited relevant data and codes to GitHub, Zenodo, NVDC human database, and possibly more importantly, CBS data sharing platform. So we developed and generated publicly available resources utilizing open life science data. So maybe the formation of this kind of positive virtuous cycle might be the key to success in open life science. So in summary, by analyzing large open data of genomic variants on both the general population from nomado and affected individuals from HGMD climber with statistical approaches and machine learning techniques, we propose a method for refining standard genetic diagnosis guidelines. And in the case 2, we analyzed publicly available disease genome sequencing data from SPARI base from a unique viewpoint, utilizing open data for annotation from ENCODE psychoencode, has led to the discovery of the role of TATs in the impact of promoter variants on ASD risk. And lastly, I'd like to emphasize the formation of a virtuous cycle of utilizing open data and developing new open resources from it will be key to success of open life sciences. This is acknowledgement. Thank you for your attention.
So we have 5 minutes left for taking questions. Do you have any question or comments? So for me first, I'm interested, very impressive results indeed, and I'm interested in the variation of TAT
Yes.
across population. Do you have any information about differences in that?
Yeah, actually, there can be variation in populations, and that might be under the control of genetic variations. But I think there is no comprehensive data of such quantitative trait loci or something. If we can construct such data resources, for example, in collaboration with IMS guys, this will be incredible resources for various field of researchers.
Well, some diseases occur for the same percentage of population across countries, but some are not. In terms of autism, do you know the risk of disease across countries?
I think there is no big differences among, for example, I'm not sure if this is a good word in the advanced countries. Actually, there is some differences between countries, but there are two possibilities. So one possibility is actually there is a difference across countries biologically. But another possibility is in some countries, ASD might be underestimated. So because the diagnosis system might be poor.
Thank you. How about questions from the audience? No? Yeah. Okay, please.
Yeah, great talk.
Thank you.
Very much impressive because, as you said, you start with the public data and so only use the electric power or something that costs. So that's how you design the project. So that's so you start with the data, the availability of data, and then think about how you kind of play with the data, or you have some specific purpose, then looking for the data availability and so which way you are doing?
Yeah, so I think both approaches can be available, but in these cases, these studies are started from specific ideas and observations.
The specific idea is not based on the knowledge of the available data, or you already have availability of the data and then thinking about some specific idea. So that's maybe.
So this study was started from my general idea that start-loss variants may not be as damaging as other types of established loss-of-function variants. And we seeked the way to statistically assess this, and we already know about this normal database, and we thought about the utilization of this dataset. Yeah, and in the second case, we first, as I told, tested this very simple hypothesis that promoter variants immediately upstream of known genes might be associated with disease, but this was not the case. So this project was once terminated at this stage. But after that, we found this interesting paper, and we analyzed the data, and we observed some results supporting our new hypothesis.
Yeah, I agree. That's the both way is possible, but maybe you have a strong kind of the capability to finish up the project, maybe. So that's and just starting with play with the data itself is not making to such kind of a great kind of conclusion. So maybe that's the most important thing is to kind of the strong kind of the driving the kind of the mind to finish up this movie. Yeah, that's very much impressive. Thank you very much for the presentation.
Yeah, and another thing is, so for example, this data set is really huge. So, but we made this data set readily available by downloading whole data set. I think that might be another key point to immediately test new hypotheses.
Okay,
Thank you.
let's thank again to the first speaker.
Okay, thank you very much for the very kind invitation. So what I'm going to talk today is a little bit of a story of a project that is the FANTOM project that started with, some idea to how to generate data to understand biology. So we started all in the 90s with the idea to do something to understand what the genome was doing. So, I joined RIKEN in 1995, and with the idea to develop technology to identify genes in a very large scale, and to provide originality, to really explore a new ground, I decided to develop methods. One method to capture full-length cDNA, essentially is to capture the full sequence of RNAs, and then a second method to map regulatory element, namely promoter and enhancers. And what I really learned from RIKEN at that time, particularly from Hayashizaki when I was a postdoc, was the importance of choosing some independent and original direction in the science. And particularly, so here is that many labs in the world were thinking about sequencing the genome, and Sanger, then MIT, that become broad later, and so on. And what will be some original project that would provide some independent data? And what we decided to instead of competing in the larger genomic area, we decided to do something complementary using the mouse, because mouse can be used to sample many RNAs, even from embryos, and also to develop methods, particularly, apart from the full length, also sequencing methods. So, and this was my postdoc work, to develop a method to capture cDNAs that reaches the cap site only. So essentially, we have full length and truncated. We cleaved away the biotin that derived from a chemical reaction, and capture only the cDNAs that are completed, where the RNA strand was protected from RNase. And then after various steps, we cloned, and then we developed additional technologies also for sequencing. A full system was like industrial production of sequence out of cDNAs and capillary sequencing done with Shimadzu. But also at the time we also appreciated the importance of full-length technologies to really discover novel genes. And then we were at a certain point that we produced a lot of cDNAs for a large number of genes, and then we found that we did a mistake. So we did not consider the bioinformatics part at that time. So we had very few bioinformaticians in our lab. So we decided to, well, let's invite bioinformaticians in the world, and let's make a project that is based on the annotation. So we decided this functional annotation of Mammalian is not a meeting where you listen to the talks mostly, but it's a meeting where you analyze the data. There was a meeting about annotation of genes, and discussion, and thinking, and and then because we had many foreigners in Japan for the first time, then we brought them to Kamakura to experience Zen meditation, and it was also quite a good thing for many international guests. So out of the first meditation of what the genes are, I think one thing that became obvious is that so if you look at the textbook, this is from a random textbook, what a gene is. You can see promoter, transcription, very simple messenger RNA. Sometimes there may be alternative splice inside, but then it produces a protein. What that data was showing was that very often we have many complex patterns. So let's say one gene, set of transcript is zero with alternate splicing, but sometimes a transcript jumps into the downstream gene. There is also a transcription from this point into the antisense region. There is also some antisense of the antisense. So essentially, the genome is all overlapping with a very complex pattern. It's not just as simple as here. So essentially, we started to see that there are many non-coding RNAs, so RNAs that do not produce any protein. Some are on the opposite strand, so we're called the antisense RNAs. And as a summary of that project, and is that this is in 2005. We expect many more genes, but the genome does not make more than 21,000 protein coding genes. But what we see at the time, we saw 23,000. There are more actually, of non-coding RNA or long non-coding RNAs. And at the beginning, we did not know anything. Little by little, many groups started to use the data because the data were open to the public. And some start to see various regulation at different levels. It was very important also that the data became public, because if so, many people could really validate, and there were many studies which have been really validating and investigating those genes. And still, for 95% of them, still we do not have actually a function. But what became clear as a summary is that before this kind of transcriptome project, we expected that the gene will occupy about 2% of the genome, and all the rest will be a desert between genes and genes. But actually, there are many RNAs jumping from gene to gene, and actually our current view of the genome is much more, kind of very crowded place where there are very few parts of the genome that do not produce RNAs, and all the rest seems to be like a very dense forest. Just for information, of course, many years ago we started to say, we have many long non-coding RNAs. Guys, please look at our data. But there were also some people that attacked us and say, but you have a lot of junk that do not encode for proteins. And then we have cDNAs cloned in the wrong orientation, but actually they were the antisense RNA. So and were a lot of reusage of our data in various other papers, but also various colleagues that could verify the all of those findings, and various other papers, including the ENCODE in 2012 that really established that there are more than 70 or 80% of the genome is actually transcribed into RNAs. So there are one important part of this is not only to make a catalog of genes, but then we move to identification of regulatory element. Let's say promoters. Promoters are upstream of protein coding genes, are upstream of non-coding RNAs, and are also produced that are also enhancers that produce RNA. So we can capture. Actually, for this we did not know at the beginning of the project. It came out during the project, but the second method is called CAP analysis gene expression. Essentially consists in capturing the very initial part of the RNA by producing a copy in cDNAs, which is usually short, sequencing by Lumina, and counting gene expression. Many copies means highly expressed, few copies means less expressed, and mapping all of this to the genome, which is also very important to identify the sequence upstream to those sequences that are essentially the promoter element or the enhancers as well. So because we capture promoter element, other groups and ourselves can do a lot of computational analysis, in particular by computing the set of transcription factors that are there and see if there is any, if we can see enrichment of transcription factor binding sites in front of genes that have the same transcriptional response. And here is just one slide to summarize the whole FANTOM 4 project, where we make where we derive a network of transcription factor binding site. And apologies, there is a name of transcription factor here, but we cannot see them. Essentially, all this network in differentiation from monoblast into monocyte centered on transcription factor, which is centered on the cage signal that identify promoters that are upregulated or down-regulated. So why, again, is important to have open science? Because, for instance, here, a group of colleagues, mostly from UK, they reuse this kind of network and then develop a method. It's called Mogrify, published in Nature Genetics 2016, but they also start a company. And the company mission is to use this information originally now in the FANTOM 4 and the FANTOM 5, transcription factor binding site, and then which transcription factor is important to convert any cells into any different therapeutic cells without doing iPS cells. So you could convert a fibroblast directly into a myoblast and B cells into macrophages, etc. And this method gives you the set of transcription factor that you should use for cell conversion. So this is one of the reasons why it is important to really have the data always open and well categorized so that they can be very accessible. FANTOM 5, this is from 2014 to 2017. We've been continuing to analyze, and actually what we've been seeing is that there are, by analyzing 3,000 libraries from many primary cells, so we start to analyze properties of promoter. We see that very often promoters have a transcription starting site specific pattern. Astrocyte, this gene which is transcribed from right to left as a single peak. Other from CD4 or CD14 or CD4 have more prevalent upstream sequences. This is all in the same promoter in 270 bases, and finally cancer cell line initiation is also here. So essentially what we see is that there are, even within the same promoters, there are different sub-components of the promoter that are active in different cell types. And if you look at this, at how many we have, calling group 1, group 2, and group 3, we have more than 223,000, which is much more than the number of the genes, which means that there are many alternative promoters, but also many non-coding RNAs, which are also have a part of this. And people have been analyzing, people have been looking at a network of similarity of promoters, and there is a lot of computation, and we don't have time to really go into the details of this. I would like just to mention about the FANTOM 5 to really mention that we also identify enhancers. Active enhancers are characterized by having bidirectional transcribed RNAs. So you can see within the space of about a nucleosome, let's say 150, 160, 170, there are two RNA going in both directions. Chromatin marks are also in agreement. H3K4 methyl 1 is higher than H3K4 methyl 3, and DNA is a sensitive site, and so on. Essentially, we can also capture enhancers, which was not expected at the beginning of the project, but it was very often you have some side discovery which are very useful. And just to mention some of the numbers, I cannot go into the details of validation, but we have been validating them, and then they validate reasonably well from cells and also down to zebrafish. Half of the enhancers can be validated by using them to promote expression of GFP in developing embryos in specific regions. Why I mention this, and this is also related to the previous talk about genetics. We are looking at GWAS SNPs, and at the beginning of the GWAS, everybody expected that GWAS will be essentially in the protein coding regions. here is the list of the diseases. We have some of the GWAS in the exons of protein-coded genes. But if you look at the number of GWAS for more diseases in promoters, or even more in the enhancers, we see that very often the human diseases are, the genetic traits that regulate human diseases are not so much in exon, but more in the regulatory element. And this is quite important to understand diseases. We are not done with the non-coding RNAs. We continue. This is more recent in 2017 with Chung that has been reanalyzed and said that there are at least 28,000, maybe there could be even 63,000 of the non-coding RNAs. And comparing with the gene code, we associate the promoter with a cage signal, so essentially cage, so we call phantom cage associated transcript. There were more than gene code. Now gene code recently has been massively expanded, the number of non-coding RNA. I should update my slides, but also we are working with the gene code as well to have the cap trapping, and so they've been also using our technology to produce a lot of CDNS sequences. So now we are producing we are at the Phantom 6, and and good part of this is not published. All of my colleagues are really working night and day to really summarize this in papers. But we are producing multi-omics, essentially cage. CVC-seq is a method using full-length sequencing, but also on the oxo nanopore sequencing. So we sequence hundreds of millions of cDNAs per each run. So essentially, we produce a huge amount of data per transcriptome that is really changing what we understand of the coding, splicing, alternative non-coding RNAs, and so on. And then we have various other technology, chromatin, screening. Paris is RNA. I don't have time to talk, but Jessica is involved in this analysis, can discuss it. And one of the key technologies is radical, which means RNA and cross-link to chromatin and the capture in a global interactome. Just I'd like to show just one slide of what we see with the oxo nanopore long read technology. Essentially, there are many points on the slide, but there are many novel genes. Just analyzing 3 libraries, there are many novel genes, many novel isoforms, a lot of non-coding RNAs. One single surprise, which I can talk about, is that enhancer RNA. Enhancer RNA, we all thought that enhancer RNA will be essentially non-spliced and short, 200-300, but actually what we see is that very often they are longer than what we expected, and very often we have 2 or 3 exons that are coming from enhancer RNA. So it is totally new understanding of what enhancer RNAs are. And this is a little bit of the preview, is all unpublished. Jessica is behind all of this, where we have a map of all the transcript in the modified Zenbu browser, but also the peak of cage with the promoter. Many more full-length cDNAs mapping here, and here is RNA-RNA interaction. So we have with the Paris, we detect all the interaction of RNAs in all the cells, which is just one system. We can see there's a very dense set of interaction. We have a DNA, DNA. This is chromatin RNA interaction. Which RNA goes to the DNA where, and which DNA receive RNA from which other place. So essentially, we have a lot of interaction with the chromatin RNA data, and we don't have time to talk about that, but just I'd like to give you a preview of what is going to come out soon. And this is a little bit, many patterns. So RNA coming from introns, distributing along chromosome, non-coding RNA providing with RNA that goes to a different chromosome and interacting. So this is a new method which was published a couple of years ago to detect interaction. RNA containing sequence of retrotransposon element that go in cis on the same chromosome, or they go in trans. Mostly with line, they go mostly in trans, and quite a lot of different pattern and a lot of analysis. And this is a little bit what we think is a phantom, which is essentially we call FANTOM when the project has produced a so complex data that a single group will never be able to analyze or store or make the analysis public. Okay, and and actually we also the data is highly reliable, so it's very important to produce a global standard for the transcriptome, for the promoter, and this time for the interactome of RNA with chromatin for screening functional RNA. And this is a little bit of picture of the FANTOM from the year 2000 in Tsukuba in summer, those 2 weeks where everybody came to Japan for the first time. FANTOM 2, FANTOM 3, 4 and 5, 6, and still going on. But what is also very important is that for many people that were at RIKEN at that time, that was very important for their career. So it's also a hub not only to exchange information, but it's also a hub to really, to really have many, to really project many colleagues in their future careers. And both Japanese that went abroad, that came back, became professor and so on. And so on. And here is a little bit of the summary, but I mentioned already those projects separately. Important here is all the data are available. This is there was Kawaji-san, and then Kasukawa is behind all of this. You can download all these data that are very well organized, and of course you can download also from public repositories. But here from the RIKEN FANTOM website, you can have all the data in a very nice organized. This is Zenbu. Jessica is behind this. This is a very popular browser, and all the data in various forms and from the file. And actually this is quite good because there are, at least in 2022, but this is quite constant over the years, about 10 million visits. And maybe one more slide, and then I'm going to skip some of them, but actually it's important. For instance, Yamanaka sensei has been using, Shinya Yamanaka has been using the FANTOM cDNAs to really have to obtain the transcription factors for his experiment. He kindly acknowledges us all the time. Those have been the cDNA have been using for the protein 3000. Yamanaka, and one more example that we identify among the antisense, some sign-up that are kind of RNA antisense that contain sign element that upregulate translation of the targeted protein, and these are probably gonna go very. I'm gonna essentially skip. Essentially, we can translate more protein from less RNA, rescuing the phenotype. This is a Medaka fish for a model of haploinsufficiency of human, with the idea that we can rescue using those antisense rescue haploinsufficiencies or the lack of protein. And finally, I would like to just show some of my colleagues there, and both at RIKEN, some at Human Technopole as well, and also, okay. Thank you for your attention, and very happy to get your questions.
Thank you, Piero. Now we can take questions. Just very informative and also historic accounts of the whole project. Do you have any questions? Well, no, for me, I'm interested in the whole FANTOM, but it started as mouse project, now it's mainly human. So when the shift happened in FANTOM 3 or 4?
We went. We shifted in about between FANTOM 4 started already to have quite a bit of human with the THP1 human monocyte cell line, and then became more obvious that although mouse is very interested to do a lot of biology, many long-coding RNAs are kind of not so well conserved. So, and if you focus on studying function, you may not find easily the equivalent in human. And given the, particularly 2013, 2014, where we realized that, having something for innovation will be important. So we decided essentially to shift. into human. Of course, mouse is fantastic for a lot of biology, but very often there is not.
The project includes many medical doctors working on diseases, or how do you communicate with those clinical people?
Yeah, clinical. Okay. At the moment, until the data that we are analyzing now, we have been working on essentially differentiating cells, IPS cells into neuro, so very basic. Now, in the current, now we are in the current phase that we are starting. We started to introduce clinical data, particularly with some specific focus on cancer, immunology, inflammation, and little by little we will add diseases as well, okay? Because at a certain point, you know, it's also the question how to budget this kind of project. Has been fantastically supported by RIKEN for many years. Now we are really squeezing the budget of my group both at RIKEN and also the budget that I have in Milan at the Human Technopole, and combining that plus the budget of a few other groups, people that volunteer for sequencing. But clearly, you know, at a certain point, having some disease we'll have to move to some disease for expanding this the number of samples that we can analyze. The mic guys are arriving, yes.
You mentioned a standard, trying to create a standardized, global standard with your data. How do you do that, especially for human? How do you determine what can be the standard, and how does that get, you know, approved or whatever, agreed upon?
Well, of course, this. I mean, it sounds a little bit ambitious when I say that we develop a standard, but. I think what at least in this kind of large projects, what you can see is that the quality between library and the type of signal is always very reliable, because it is done having following high, reasonably strict quality control in the preparation of the data, and produced by the same lab, run with the same pipeline. So essentially, we know that if there is a bias, it's going to be constant, and probably you can there's very little variation between the quality of the sample. But then if you pick up a sample that are not a good quality, we usually eliminate. If you go in public domain, you see a little bit of everything from different quality from many different groups, so it's a little bit of critical mass. And then, well, if the RNA is complete, is a full length, we compare full length sequencing with CAGE, and CAGE, the signal of CAGE is very clean. We identify promoters. For instance, for determination of the transcript is a little bit more complex, but we've been analyzing internal priming for the gene models, and can filter that. But it's the aggregation of several qualities, and all the questions that all the requests of the reviewers to validate the existence of the RNA, the presence of the promoter. What we call promoters are really promoters or not, and or an answer. So this is a larger data set that survives kind of a large number of requests from reviewers, and then it is public, so everybody can reuse. And they can correct us, or can modify. They can reinterpret the data. So, and yeah, of course we are always open. No, we are always careful to not to overstate.
Okay, thank you.
Right. Thank you.
Good afternoon, everyone, and thank you very much for inviting me for this interesting conference, symposium. I'm an astronomer, so some of the part of your presentation. I couldn't understand the wording things in English, but maybe my talk, you might feel similar things, but my talk will be more on the overall things rather than the specific science itself. Well, actually, I'm the astrochemist and studying radio astronomy, so that my talk will be dominated by the knowledge surrounding me, so that insight and case studies from experiences in astrochemistry. Well, before going into the open science in astronomy, I just want to it's a bit dark. Sorry, remind you that the astronomy historically developed as a practical discipline closely tied to politics and agriculture. However, as you know, the modern astronomy primarily focuses on the understanding the origin of the and evolution of the universe and ultimately our origins. So that our curiosity is based on not like making our money or contributing to the humanities, not like that. Rather than that, we based on the pure curiosity to know our world. However, the astronomy often giving you the new ideas to open a new field, so that indirectly we are contributing to the development of science, but not directly related to that. That's the point. So I think that is another key point why astronomy is successful doing open science, because it's not directly related to the benefit to the somebody else. Okay, so in astronomy, we have two types of interests. One is structure formation in the universe, and the other is the evolution of matter. Well, in both of the cases, we must to know what's going on there. That means we need telescope to know that. Not only with the theoretical calculation, we cannot know the universe. We have to observe it. To observe, well, depending on material, I would say depending on the matter, the frequency, there is a difference in frequency. If you want to know about inside the atoms, you have to go to the higher frequency or shorter wavelengths. And if you want to know about the molecules, you must observe the radio wavelengths. So that depending on the frequency, our community is somewhat divided. So that the high energy people is in this end and the low energy people in this end. Well, despite this, the separation of the field, we are doing nicely the collaborative works by using the same format or by comparing a data set. So how? Well, before going to the detail, I just want to introduce the best case, I would say, the archival data set in ARMA. ARMA is a large millimeter, submillimeter array located in a 5,000 meters high altitude in Chile, the desert. And then, well, this is a common users facility developed by many countries. You see the flags here. So more around 50 countries contributing to develop this or operating it. And so that this is totally the international common users facility. And then providing the only one radio astronomical data in the world, because this is the best telescope in the world. Because of reason, you see, this is the competitive ratio in each cycle. Cycle 0 is the starting time of ARMR, and then each year we have a call for proposals to distributing the observing time. So right now we are in cycle 11 or something like that, but as you see here, mostly the competitive ratio is an average, somewhat like 5 or something like that. But please remind that this oversubscription rate strongly depends on the time of the sources, and then also the antenna configuration, so that some popular the antenna configuration with the famous stars case, the competitive ratio easily exceeds 30. This is just the average. So that you can easily imagine how difficult to get time to be observed. So that means after the observation, everybody waiting the data to be opened. So people want to look at the data because only one group can get the data for instance, one configuration with a certain time. So that the archival data is awaited. Actually, as you see here in the cycle 0, it was the competitive ratio is something like 10. And only 18 proposals are accepted from East Asian field. So it's super hard to get it. Because of a reason, ARMA, well, actually the providing science archival data, and also it's very precious data, so that the ARMA want to provide quality assured data on the archive. This is, I think, the key in this facility. Always, after the observations, there is a quality assurance. If it doesn't passing through, it goes back to the observation again. And then finally, if that pass through the quality, then it will be delivered to the proposers, and then after 1 year or the half year for the director's time case, it will be open to the community. Then anybody can download, even you can download the data, ARMA data. And also there is the common software to analyze the ARMA data, so that anybody can study the stars with free, I would say free money. I mean, there is no charge to get. And even for the observation, there is no charge to be observed. You don't need to pay for that. It will be observed when it is accepted. And that is done by a peer review system. Well, this one is actually the ARMA Science Archive. They showing the kind of map of the sky, and then the circle is the observed place by ARMA, for instance, and then here is the frequency range, and some yellow part is actually the observed one by ARMA, and then you can see when you click it, you can highlight the project here, and then when you click each of the projects, you can download the data. So this is really a sophisticated one. And when you click some part, you can see the roughly analyzed data here to check the data quality as well. And then all the information about the proposals or abstract or resolution, frequency or the weather condition, all the metadata is included, so that everybody can study it. Well, because of this nice archival system, people start to use the non-PI data. I would say non-PI data means it's not the data taken by that person, but taken by somebody else and then downloaded from the archive. Well, if you look at the nice statistics, well, you can see in a first year, the observation programs are used. In one paper, one observation has been used. But in the later stages, you want to compare the results with others, so that the number of programs will be included which is used in one paper. And then the similar things here, you see here is how many papers have utilized a single observation program. So that that means the first the year's data has been published in many papers, so that the old data is used so frequently. That means the archival data is working nicely, and then it's linearly increasing. Then you see here is the number of publications using archival data. Then green is pure archival data publication, and then blue is PI data plus archival data. And then here is the pure PI publication. So, and then archival data publication increasing. So anyway, we are successfully using the archival data. Then after the accumulation, probably someday we might have the more publication from archival data. But there is, of course, some issues. We see nice results, and then right now 40% of paper uses archival data, and then a certain percentage of the paper even producing the high impact results like published in Nature. But you see, right now we have around 2 petabyte data, and then each year we increase the 200 terabytes. Well, this is just the packed raw data. So if you want to distribute and then to start the analysis, it's 10 times more required. So that right now the issue is the size of the data in ARMA. Well, this kind of effort has been done not only by ARMA, but also by several other, or most of the radio telescope, or the I know that at least the infrared telescope. In this case, this is the data archive in a very large array, VLA, in NRAO, American Telescope. Maybe some of you know this, the movie. It's an old movie, The Contact. The Jodie Foster is hearing the radio things by commuting here. But right now, we don't need to be there. We just use the archival data. Or here is the brand new telescope, James Webb Space Telescope, and then downlinked all the data is listed here, and then most of them are immediately open to the community, and then you can download. Only a little you can see the before the priority time, so that means the I think at most 1 year the PI can use only by him or herself. So you see where this kind of database provided by observatory is somewhat reliable, because observatory secure the data quality and then controlling everything. But as you see here, for instance, even here, where this photo comes from, where this line information comes from, all these are based on a small databases. ARMA 2, ASA 2, actually just uses a small database in outside of ARMA. That is the little issue recently we are facing on, because for instance, for the photo I showed, that is coming from the database of SIMBAT database. This one is actually the historical one, and then it's a kind of set of identification, measurement, and bibliography for astronomical data. And then this was developed by Strasbourg Astronomy Research Center. It's not by observatory itself. One institute actually contributed with a voluntary mind, because they wanted to have it, and then started to open to the community with free of charge. That was wonderful, because as you see here, this providing us to compare the different frequency or different wavelengths data in the same position. You see here is a pillar, and then the different nature we see. And sometimes we can understand there is no data. That is also helpful because we are trying to get the new data. So this is super helpful to find a target to be observed in a new frequency. Well, another things, well, this one is a kind of successful one because right now, this one is recognized to be super important database, and then even financially supported a little bit. But for instance, if you go to the lines, molecular lines I showed up, there is many issues recently found, actually. For instance, CDMS, Kelun database for molecular spectroscopy, it named as Kelun database so that they operated by Kelun University in Germany. However, this is mostly operated by one researcher. And then he's gonna retiring within a few months. So you can understand how severe the issue is. This is most reliable molecular line database. Then you see a JPL that is funded by NASA, and then looks like the institutional funded one. However, this was again checked by one specific researcher. Because of reason, recently we found that many problems in JPL, inconsistency between the data and the actual intensity or the frequency. As you see, here is a JPL, and then here is the actual data, and you see the difference in the intensity, for instance. And then JPL just telling us that this is our data is not reliable. They are saying it's honest. But they doesn't giving us enough information. And then some of the database just uses all these results without thinking about the reliability of the data. That is very dangerous. However, ALMA uses this Spratlog. So there is a bit fragile situation in the background. But some of the French group actually started to follow up these two because they noticed that how important these kind of things is. And then sorry, I just want to skip this, but all those databases are actually important. However, the current issue to address is that we have to think about the reliability of the data or sustainability. Because of the one contributor case, we need the person who will continue to do that, or even budgetary support. And also the reliability totally depends on one or two persons. That is very dangerous. And then for the calculation, situation is a bit more, better. Because right now, we have some common analysis tool in radio astronomy. So we can use the same software developed by several observatories so that this is reliable and then always updated unless those observatories exist. And then for the more secondary analysis tool, AstroPy, this is also the common development tool for the software. So that we astronomers, each of us can contribute to develop the software by using this AstroPy. And then it will be implemented to the Jupyter Notebook, and then we can do analysis. This is really the kind of collaborative developing tool, and then open science things. But all these are kind of just providing a tool, and then it's not directly telling, providing you the scientific result. So that's actually why it's successful. Because everybody just contribute, and then can use, instead they can use the other people's software or some feedback they can get. So it's very nice. Then finally, for the advertisement, maybe you know this site archive. It is not only used in astronomy, but also in many other fields. because this archive system is developed originally by physicists, and then astronomy is closer to the physics field, so that's originally we used this archival system. In this system, well, at least in astronomy, you see the black part here. A certain amount of paper continuously published in the archival system. However, we are using this as an advertisement of accepted papers rather than the papers which is not yet accepted. So most of the case they are publishing here, but after the acceptance or almost accepted case. And then that is actually, you see here, citations for this archival paper is super low in astrophysics because most of the citations goes to the original journal. So that is the way we astronomers use this archive. Because otherwise, some strange papers appears in the astronomy field because, you know, the astronomy field, there are so many amateur people who loves cosmology, who loves astronomy, sometimes providing some weird papers, not reliable one appears as well, so that so the important point is that we see, which is already accepted or almost accepted by a peer-reviewed journal. Otherwise, we cannot qualify or we cannot believe that those results. So anyway, there are several issues and also the good point here in astronomy. The good point why we success the open science. The good point there are 4 I listed up, but the 2 of them are most important. One is we are using international common users facilities, so that all of them want to get the data from the worldwide. And then open sky policy, and then anybody can apply with free of charge, and then that should be distributed again to the others. And then the other one is here. I will explain that the standard data format, FITS file. In astronomy, started from 1979. In that era, we started this FITS format. I mean, all the final products is in a FITS format, so that we can easily compare all the astronomical data because the format is standardized. So well, it's a long history, and in 1998, I think almost the earliest case to use, or maybe some the spectroscopy data they had a standardized format. But anyway, still we are using FITS file, so that helps us to start the things. But there are several. Finally, this is the final page. Sorry, there are several difficulties to continue this successful open science. I mean, for the first one, common users facilities one, we are doing a peer-reviewed system for the proposals, once per year, and super competitive. That means when you submit a proposal, idea has not yet been observed. I mean, but the reviewer can notice the knowledge. And then if the proposal has been declined, and then next year, one of the bad reviewer submitted a proposal with the same idea, with a little model, then if it is accepted, well, that's a kind of success of the steering person. That kind of things can often happen, so that we are super nervous for the, opening our ideas to the others, because otherwise people can easily get the sky is there, the same facility, everything can be stolen. It's a bit dangerous situation. And also in a peer review system, of course, if all the researchers are kind of having a very good mind, it's okay. But depending on the region, there is the trend of research. For instance, in Japan, this kind of trend of research exists, but it might not be evaluated highly in other countries. That kind of things can happen. In that case, well, if the number of reviewers are equally distributed, we can fairly do the reviewing. peer-reviewed things. But if it is dominated by the one region, for instance, the European region, it's been hard to be accepted a Japanese proposal because not all the reviewers are the good person. I mean, they want to get the data for the popularism in popularity, understandable things for the, for instance, in a European field, or vice versa. So that sometimes the minor region, the idea, will be destroyed. I mean, disappeared or just stolen. So there is anyway the issue in this system. And then another one is, as I told you, that there is an issue for the sustainability and reliability of the fundamental database, which supports the observations or analysis or data analysis, the data interpretation. And of course, the increasing data sizes that might be solved if we put a huge amount of budget. Also, in some cases, in an observatory, the person who contribute to the developing a database sometimes not enough evaluated as a researcher because they contributed their half time to make a database, and then only half time could be used for the science. In that case, that kind of person, of course, might struggle to get a new position to be promoted. Well, within observatory, there might be, but if the person wants to go outside the observatory, there is an issue. So that anyway, there are so many things actually appearing. Maybe because of the success of the open science, we are facing on this issue. However, we have to think about how to solve this issue, especially this one, sustainability of the local data is super important. If some point, for instance, the other countries' data is close to, for instance, one of the country, the researcher in that country cannot do even more continue the research because of the key database is close. So that kind of risk we have. Anyway, there is so many risks and difficulties. However, the people gradually started to know. So that I think in the near future, we will think about the sustainability of the small databases in the background of the big archival system. So thank you very much.
Thank you. We can take a few questions. Please.
Okay. Do you want me to use the mic or not?
If you say in large voice, I think it doesn't matter.
Okay. Thank you very much. That was an excellent talk. A couple years ago, I was at a NASA big data conference, and one of the topics they brought up was about the data sizes and the fact that downloading these files is so huge and you need big computers with big memory. Is that a problem that you notice also in the field?
Yes, you're right. Especially for ALMA, for instance, even for the individual observation programs, you need hundreds terabyte data to be downloaded. So that the laptop is very, you need a storage or the desktop computer to analyze. So that some people or some the country people are facing on the difficulty. However, to overcome that, ALMA recently started to provide the roughly analyzed data, or some of the already, I think, up to cycles 5 or 6, all the reduced data started to be provided. In that case, they can easily access to the data. And then if there is more analysis they need, they can send a help desk ticket to the ALMA. So ALMA, I think, mostly solve those issues, I guess.
And are the other observation platforms also starting to work in that direction?
Well, some big group following that way. Like ALMA is operated by NRA or NLJ or those facilities, the institute. Those is a common in Berlin or any others, so that we can use the same knowledge.
Thank you very much.
One question. In which country the ASA archive is located?
We have 3 mirrors because the ALMA is mainly operated by 3 regions, Europe, North America, and East Asia. So that here is kind of share. The North America, East Asian, Chile, the share of the time, and then depending on the budgetary contribution, personal contribution, this share is decided. So that at least these 3 we have mirror site for the archive, and also Chile as well, of course, the telescope there.
So original data measured in Chile are sent to...
Yes, yes, yes. All these, the mirror site.
I see, I see. Thank you. No more questions from the audience? Well, we have a coffee break next, so let's discuss.